accurately reflect the work that we were doing. So the CDWG is chaired by Mike Fries, Heather Mefford, and Scott Myers, and our PI liaison is Crystalise Martin. And currently we have 2 Gene Curation Expert Panels, or GCEPs, and 2 Variant Curation Expert Panels, or VCEPs. The 2 GCEPs are both approved and actively curating, and I'll give you some sense of their progress momentarily. Our 2 current VCEPs are focused on brain malformations, specifically the mTOR-PIK3-AKT pathway and Rett Angelman-like disorders. The brain malformation group, which is one of the NICHD U24s, is in the process of finishing their initial variant evaluation pilot, while the Rett Angelman group is currently in the process of selecting the variants to begin their initial pilot of their rule specifications. Each of these groups holds independent working meetings throughout the month, but then once a month we all come together to check in and discuss any cross-EP issues. Next, please. So as I mentioned, both of our GCEPs are fully approved and actively curating, and both groups are working through lists of genes that are currently on clinical testing panels for their respective indications. Within the broad indication of epilepsy, encompassing both broad panels and panels targeting specific seizure types, there are actually over 1,000 genes on current testing panels. And we're starting with a subgroup of 45 of these identified by our experts as being kind of the most important that they wanted to do first. To date, we've evaluated 36 of those, 36 unique gene disease pairs. Most of them have been definitive, though 10 have been limited or disputed, highlighting the importance of this work. Next. Our ID-Autism GCEP is evaluating genes currently on clinical testing panels marketed for intellectual disability, developmental delay, and or autism. And there are currently over 700 of those genes. This group is initially focusing on the top 100 genes that appear on 25% or more of those testing panels. At this point, we've evaluated 78 of them, and similar to the epilepsy group, most of them are definitive as expected, but there are a handful that are limited or disputing evidence. Next slide, please. So in order for you to better appreciate some of the challenges we face when we evaluate these types of disorders, let me give you a little bit of background about what we mean when we say neurodevelopmental disorders or developmental brain disorders. So we've come to think of these as a spectrum of disorders that can manifest as features in a number of different domains, so cognitive domain, motor, neurobehavioral, and neuroanatomical or neurophysiological. Individuals with neurodevelopmental disorders may manifest features in one or more of these domains, and the constellation of the particular features that they exhibit is what leads to their specific clinical diagnosis, such as autism or intellectual disability or schizophrenia or what have you. And these types of disorders can be caused by a number of different factors, including both genetic and non-genetic causes as shown at the top here. And in any given case, there can be more than one of these factors acting together, and any or all of them can be compounded by environmental influences. Next, please. So it's not uncommon for pedigrees in neurodevelopmental disorders to look like this on the left. Various different clinical diagnoses scattered throughout the family. In this way, they can sometimes be more complicated to evaluate than your more traditional specific Mendelian phenotypes, as some of these clinical diagnoses are relatively common in the general population, like their anxiety, depression, etc. And it can be unclear whether they're part of the disease spectrum or if they're phenocopies. As an aside, there's also the issue that some of these things, when you're taking a family history or looking at a family history, some of these things are anecdotally diagnosed by individuals or family members themselves rather than clinically diagnosed by professionals. As genetic testing within these disorders became more widely available, however, we began to realize that these seemingly distinct clinical diagnoses were often caused by the same underlying genetic etiology. So now you can see this is the exact same pedigree, but the affected individuals on the maternal side of the family are all manifesting different features associated with FMR1 expansion. Next slide, please. And further complicating the evaluation of neurodevelopmental disorders are issues surrounding variable expressivity and seemingly reduced penetrance. So we know that parental neurodevelopmental performance kind of sets the threshold, if you will, for the expected performance of the offspring. The child of genius parents is probably more likely to be at a higher threshold than the child of parents who have had neurodevelopmental deficits. So if, for example, a child has one or more of the various genetic hits, if you will, associated with neurodevelopmental disorders, that performance level is expected to shift downward. How far it shifts and in which domain depends in part on the particular genetic variant and whether or not this shift lands you below the threshold for diagnosis, which is, you know, that red dotted line down there for, say, intellectual disability or autism or what have you, depends in part on your starting threshold. So you can see in both individuals here, the presence of a genetic variant results in a downward shift in their ability across domains from expected, but this shift doesn't always translate into a clinical diagnosis or even the same clinical diagnosis. Individual A would perhaps be diagnosed with an intellectual disability, while individual B may perhaps be diagnosed with autism. As you can see, some of these downward shifts still leave the individual within the average range or the subclinical range. And that contributes to the perception, you know, that we see sometimes that some of these variants have reduced penetrance or that certain individuals are quote unquote unaffected. Next slide, please. So to summarize, the nature of our disease area leads us to the following challenges. We're dealing with a disease area in which certain presentations are relatively common in the general population and for which there is extreme genetic heterogeneity. When we are evaluating cases and variants, we really need to pay attention to how thoroughly the individual has been evaluated and how sure we can be that a variant that we're looking for is the variant of interest, that there's not other things that haven't been ruled out. There can be great phenotypic overlap between our conditions. We frequently see intellectual disability, autism, and or seizures in many of our gene disease evaluations. Both of the GCEPs carefully pre-curate their genes in order to be very clear which disease we're evaluating and which phenotypic presentations we'll accept as contributory for a particular curation. And due to this overlap, our CDWG has actually created our own Mondo term, complex neurodevelopmental disorders, to describe those disorders that don't otherwise have a recognized name, in which more than one neurodevelopmental disorder is present, and Andy will discuss this later. We also stay in close contact with each other through the CDWG in the event that a curation may be more appropriate for one of the other groups. As I mentioned on the previous slide, the issues inherent in pedigree evaluation, variable expressivity, apparent reduced penetrance, and situations that look like they could be non-segregations. We need to evaluate those carefully to determine if we really feel that we can say they represent true evidence against an association versus no clear evidence either way. And further, some more mildly or subclinically affected individuals can make their way into population databases, so that always needs to be kept in mind. And finally, while we're lucky to have some very large cohorts for certain indications, such as autism, they are often used and reused over multiple publications. So we really need to be careful that we're not double counting cases. Because these cohorts are so large, it can be difficult to assess the data for any single individual. So, for example, it might be hard to know if other variants were identified in that particular individual. So at this point, I'll turn it over to Andy, who will walk you through a particularly difficult case within the ID-Autism group that hits on some of these issues.
Thanks, Erin. If everyone could just please mute themselves, there's a little bit of background noise. Thanks so much.
Yes, thank you. So as Erin mentioned, and Jenny mentioned, I'm going to be going through a complicated gene disease curation that we did in the ID-Autism group. And this gene is the RELN gene, which we ended up curating for complex neurodevelopmental disorder. And sort of the interesting thing about this gene is that when we were first sort of curating it, or at least when I was first curating it, it looked like the gene was going to end up being definitive just based on the wealth of evidence that's available in the literature. And then as we sort of took a deeper dive into the actual information that has been presented, we actually ended up disputing the gene-disease relationship. So I'm just going to walk through sort of the entire story of our process and some of the issues that came up, and and then maybe if we have time, we can discuss some of those issues. I will note that this process actually started with a pre-curation in September of last year, and when it was first presented in February 20th, that was when we had thought of it as definitive, and then we discussed it on 2 calls in March and had an offline discussion, and we just finalized it last week as disputed. So just to give you an idea of sort of the arc of the timescale. So the interesting part. So just a little bit of background on RELN. So RELN is expressed in the brain during early development. It is thought to activate a signaling pathway that triggers nerve cell migration, which then facilitates axon and dendritic extension and regulates synaptic plasticity. RELN has been studied for over 20 years and has been a possible gene involved in neurodevelopmental disorders. Some of the evidence to support that is as I mentioned, the expression evidence. So in the GTEx database, we've seen that this expression of RELN is restricted and high in the nervous system relative to all the other tissues in the body. So it would make sense that it may have a role in neurodevelopmental disorders. On top of that, it has been asserted in several different databases, including OMIM and Orphanet, for different neurodevelopmental disorders. And when performing the pre-curation, we found that it was described in most of these databases as a Mendelian gene for autosomal recessive lysencephaly, as well as a susceptibility gene to autosomal dominant temporal lobe epilepsy. We also observed that there's an extensive wealth of literature implicating this gene in autism, which was autosomal dominant inheritance, and that the gene has also been altered in patients with complex phenotypes such as myoclonus dystonia. And then mouse studies have suggested that the gene may have a role in schizophrenia and or bipolar disease, but there haven't been any human variants that have made this assertion. So just to summarize what we sort of concluded from the pre-curation with all of this, so per the OMIM and Orphanet literature, RELN's been associated with the temporal lobe epilepsy as well as the lysencephaly and autism. And since the lysencephaly gene-disease relationship follows the separate inheritance pattern of the autosomal recessive inheritance, and it has a different phenotypic spectrum, we felt that this should be assessed as a separate and independent curation, so we split this out and sent this to the ClinGen Brain Malformations Gene Curation Working Group for further assessment at a later date. I'll also note that initially it was thought that the seizures that are observed, the familial temporal lobe epilepsy phenotype could be considered as part of the CND spectrum along with the autism/intellectual disability cases. So we had opted to proceed with the complex neurodevelopmental disorder as a lumping term. So as Erin touched upon in a little bit more detail, just the overview of the definition of complex neurodevelopmental disorder that we were caring for is just a disorder that involves more than one phenotype associated with the central nervous system, including but not limited to ID-Autism and seizures. So for the purposes of this curation, we chose to curate cases that had neurodevelopmental disorders that had a neonatal or childhood onset. And when we took a closer look at those adult onset epilepsy cases, we actually found, or the epilepsy cases, we actually found that they were adult onset, as well as the myoclonus dystonia. And so we found that these were not scorable for this relationship as they sort of fell outside of the clinical spectrum that was more consistent with the gene. So when we look in NOMAD as the next, or EXAC as the next sort of spot in the pre-curation, we found that this gene is not constrained for missense variation, which ended up playing a sort of a big role in making us uncomfortable in awarding default de novo points to some of the variants that were missense and de novo and didn't have any further evidence for pathogenicity. And while this gene is constrained for loss of function with the PLI score, we cannot assume that loss of function is a mechanism for this particular autosomal dominant CND gene disease relationship without the supporting functional evidence. So that sort of put a little bit more of a, made us in more of a critical mindset or critiquing mindset when we were looking at these associations. So now I'm going to go through the sort of the first pass that I had taken and we had taken when we initially started the curation for the RELN gene. So as we normally do with the gene disease curations, we typically start by trying to count all of the case level evidence and adding up, missense and loss of function and de novo variation. And we initially were trying to incorporate the temporal epilepsy cases, the myoclonic dystonia cases, and then the more controlled autism spectrum disorder, intellectual disability, and developmental delay cases from some of the other smaller sequencing studies. And then we found a large sequencing study paper that presented a wealth of sort of de novo variant information and sort of gave us that wealth of information that we were looking for. But then we took a closer look at it. But I will get there when we get there. So to start, I just want to highlight the first paper that I curated. So I initially went to this paper because it had extensive family information for the autosomal dominant lateral temporal epilepsy. So all of these families are the large families that have shown to have some segregation of heterozygous missense variants with the latent temporal epilepsy. And these different colors are actually, some of the various forms of the epilepsy, which sort of hints at the wide phenotypic spectrum that we maybe could have observed with this gene. And so the important thing that we thought about this was that if you look at all the figures at the bottom, you can see that there's actually a lot of family members that do not display a phenotype despite carrying the variant, showing that they have reduced penetrance. And given, as I just mentioned, that the phenotypes are very different from the clinical spectrum presented in the majority of the patients with a complex neurodevelopmental disorder in this gene, so mostly largely autism and intellectual disability, these cases were not deemed reliable for scoring, and we decided to omit all of these cases from the curative phenotype in the complex neurodevelopmental disorder. And I should note that none of these variants had any further evidence for impact, so they weren't really scoreable on their own. So these next families are actually from the same paper, and they're just more families that the group had decided that should be separated from our ID-Autism group's curation of this complex neurodevelopmental disorder in order to give us a more specific spectrum of disease. The epilepsy group may revisit this at a later date, but we felt that this didn't match the RELN phenotype that we were trying to carry. So the next cases are the myoclonic dystonia cases. So this was the only paper that asserted for the myoclonic dystonia. And it's sort of interesting that all of these cases, despite having sort of a pretty consistent phenotype in a lot of ways, the onset was variable, and the allele frequencies of the variants that were asserted in these publications were actually above what we felt comfortable with scoring, and some of them were actually above a lot of groups and expert panels thresholds for BS1 and BA1 and could not be scored. So we did not score these cases. Let me move on to the next sort of segment of the cases we have, sort of these papers that are some of the smaller cohorts that sequenced patients with intellectual disability, developmental delay, or an autism. And so what I mean by the smaller studies is that they're not part of sort of the big cohorts that Erin was mentioning that are traditionally like sequenced and resequenced or re-reported. And so these are, I guess you could say, novel cases for lack of a better term. But what we noticed with these is that a lot of these cases, while having a neurodevelopmental phenotype, the NGS panels were small and they weren't comprehensive for the genetic heterogeneity that is observed in these disorders. And given that they were just missense variants, we felt that these really didn't have enough evidence for pathogenicity and that we couldn't score them any more than 0.1 points if we were going to even give them that. And given that this was sort of a consistent trend, we found that we could not score any of these, sort of looking back at it. But one of the other interesting things is that that I do want to make note of is that some of these patients, like for example, this patient was sort of part of a large cohort and the variant was reported, but it doesn't really say which patient corresponds with which variant, and they really don't provide any further information about who the patient is relative to the variant, and they're just it's only reported as a variant that was identified in a high-risk autism spectrum disorder cohort. So we couldn't score this variant in addition to the fact that it was not functionally assessed or any other supporting information. And then this was another small panel, but there have been RELN variants that have been de novo that have not segregated with the disease. This is an example of a patient with a sibling who had autism spectrum disorder, but this loss of function variant in SCN2A was the segregating and causal variant. Okay, so now we're going to get into sort of the large autism spectrum disorder cohort study. So this paper here with the screenshot on the right is a large autism spectrum disorder 2017 study that investigated a number of gene associations with autism spectrum disorder, 91 genes to be exact. Many of the variants were reported as de novo, and therefore they would have helped us reach a definitive classing very quickly. And it was, at first we thought this was nice. This is reviewing and reusing a lot of data from a lot of autism spectrum disorder studies that we hadn't scored yet. But we had to be careful not to double count the RELN cases. And just to give you an idea of how that can be sort of difficult, these are all of the sequencing cohorts that were included in this study. So we had actually initially scored, or I had scored variants from the Derubius and the IOSFAV 2014 papers as independent cohorts, and then I noticed when we were scoring the Stessman cases that they were actually double counted. Additionally, it's important to note that a lot of these cohorts have a lot of information in the supplements that sort of gets lost. And again, sort of the who is who, which variants are associated with which cases, and this can often lead to ambiguities in what type of testing was performed for each of the cases, what other variants may have been detected in those cases, and who the affected individual in the family is. And so we felt that the only way to sort of score this paper, the Stessman paper and all the data that was in it, was maybe we should lean more towards a case control. But before I get into sort of how we did that, I'm just going to do a brief overview of all of the cases that we do have, just to give an idea of where we were coming from at the beginning when we felt that this might be definitive. So we had the 7 families in the Jazzo et al. Paper that had the autosomal dominant temporal epilepsy and the missense variants that were segregating, albeit with incomplete penetrance. We have the 3 families that had the myoclonus dystonia that we had sort of disputed for the population frequency. We had 4 missense variants that had no phenotypic data or evidence of pathogenicity. We had 1 missense variant that only had those 44 genes sequenced. But then in these sort of larger cohorts, we had 7 de novo loss of function variants that were identified by whole exome sequencing. But these were actually not known to be confirmed by orthogonal testing. And this is, again, sort of one of the faults of the supplement is that they have a column that says confirmed variant, and a lot of these that we were trying to look at with the RELN were called unknown, so we weren't sure if they were confirmed via orthogonal testing. So we definitely didn't feel comfortable scoring these as case level, but we thought about maybe trying to get evidence from the case control. There were 4 de novo loss function variants that were part of the Stetson paper, I believe, that were novel cases. And since this was sort of a case control paper on its own, like sort of by nature, and that they were trying to identify as many of these 91 genes as they could that would have associations with autism and intellectual disability, we felt that these should definitely be looped into a case control. And then the last thing that we did find was there was 1 de novo missense variant that was found to have that non-segregation in the sibling-like disease. That was that SCN2A case. So when we look at all of the case level data, we came out with 0 points in the hope that we would maybe be able to score some of these cases in the case control section for assessment. But just to give you the brief snapshot of what this looked like when we tried to score it the first time. So we had much more than 12 points of genetic evidence from these de novo occurrences and the segregating missense variants. And then we had actually a lot of experimental evidence before we took a closer look at this as well, because there has been a lot of functional and expression studies indicating RELN's role in neurodevelopment and neuronal migration. And there are animal models. There's actually 1 animal model, but there's a lot of papers describing a heterozygous mouse with RELN variation that recapitulates or has had mixed results in recapitulating some of the behavioral or learning deficits. But there were sort of enough studies that when we aggregated all of the evidence, we could actually max out the functional evidence and the experimental and model evidence. However, after we got to this point, we had all of our concerns that I sort of outlined when we were looking at it at a case-by-case basis, and we felt that given that there was all of this discordance between the studies experimentally as well as the lack of pathogenicity for the de novo variants, other than the fact that they were de novo, we felt that sort of moving towards solely the case control viewpoint would give us a more conservative and accurate representation of the evidence. So just to provide one last overview of the traditional scoring, so to give you sort of the final count, and some other thoughts. So there are at least 25 probands that have complex neurodevelopmental disorders and at least 10 publications with RELN variants. And though many of them have been reported, there's little evidence to support their pathogenicity. We do not know exactly the molecular mechanism for autosomal dominant RELN variation. The de novo RELN variants have been reported as part of large sequencing studies. Many of them are part of these large sequencing studies, and then several publications have repeatedly used these large sequencing cohorts in their analysis, which sort of muddies the waters. And then in order to not conflate the GCEPs classification, at this point we decided to score 1 study, that assessment study, that combined all of the large sequencing cohort data as a case control. So when we look at just the case control evidence, I just want to note that there have been 11 papers that have done some statistical analysis analyzing cohorts of autism patients where RELN has been investigated. And since all of the reported variants had reasons to be downgraded or discounted, we chose to just score these 11 papers. However, when you look at the papers, we found that I guess I'll start at the beginning. So the first paper, this particular et al. 2001 study, identified that the CGG repeat in the promoter region of RELN was significantly associated with autism. So therefore, they stated that RELN was a candidate gene for autism in 2001. This sparked an interest in the gene, and many papers tried to replicate this result as well as find novel variants like those presented in the case level section. However, many of these publications were not able to replicate these results, i.e. these studies right here, and they did find novel variants like those presented, but when they tried to replicate the results in the statistical methods, they were not able to consistently demonstrate this gene-disease link. Many of these studies had investigated the CGG repeat explicitly, but they also looked at some of the other variants in RELN. And there was one study that did find a significant association with RELN, but they didn't actually have any controls, so we could not score this paper here, the Scar et al. 2005 paper. And after all of this contradictory evidence, there were still two papers that did assert the significance for the CGG trinucleotide repeat. However, if we look at the data that's in NOMAD, for RELN, we see all of these CGG repeat variants, and we see all of the homozygotes and the high level of presence of these alleles in the general population. And we feel very comfortable that we can conclude that these CGG repeats should be considered benign or at least not scorable in any way. So now if we go back to these Stetson papers. So as I described at the end of the case level portion of the presentation, many of the cases that were previously published were compiled into this large case control analysis. And there were some new cases that were added via this publication's own study of 208 autism genes. And they were trying to assess which genes had a difference in the association with intellectual disability versus autism, which is this statistical analysis that's highlighted in the blue box, as well as which genes had likely gene disrupting variants whose frequency was significantly increased in cases of autism slash intellectual disability versus healthy controls, which is this the red box. So they found that RELN did have a significantly higher rate of de novo LGD variation in autism patients relative to the intellectual disability patients, as this p-value here, which I believe is 0.001. But they did not find that there was a significant difference in autism patients versus controls. So they did find that there may be some level of significance of this gene's association with autism, but we felt that it wasn't necessarily scorable. This type of study is actually not supported by our classification matrix at this point, so we really couldn't use this level of evidence. But given that there was sort of all of these cases, we felt that maybe we could do our own case control study in an effort to try to better understand the relationship between the RELN variants and the cases versus the controls. So we tried to use a chi-squared test, and we found that there's actually a significantly higher amount of the variants in the patients included in the study relative to the controls, which were exact controls. So since these controls are from an exact cohort that was not ethnically matched or age matched, and may not have been thoroughly assessed for the disease, we felt that we could maybe score this, but be very conservative by scoring it as 1 point. However, we soon discussed with the experts, and we realized that this really wasn't a statistically robust, sound way to provide support for this association. And this led to a lot of discussion and second guessing of our methods. But fortunately, the experts actually found a newer paper that has been posted on bioRxiv that provided some clarification on what we should actually do with the specimen paper. So this paper by Barrett et al., 2017, found that many of the statistics performed in Sussman et al. Were actually incorrect. And in finding this, they prompted Sussman et al. To provide new supplementary tables with the corrections. And these indicated that RELN was actually not significantly associated by any metric, be it autism versus intellectual disability or autism and intellectual disability versus healthy controls. So that made us feel more comfortable about not scoring any of the case control evidence. I will note that the intent of this paper was not to be contradictory to the RELN complex neurodevelopmental disorder disease assertion, and it wasn't to directly dispute it, but it was mostly just to display that there were incorrect statistical analysis, which then prompted sort of all of this re-review of the statistics. So the other two important things to note is that this paper actually didn't come up in PubMed at all. It does not have a PMID. The only way to find it is actually by performing a Google search, and it's not associated with the Sussman paper in any way at all via the search. So this is just sort of an alert for biocurators that sometimes, even for gene disease curation, it's always good to do a broad search on a larger search platform like Google, in addition to any PubMed searches that you might do. So this is just a quick slide on the corrected table from the Barrett and Sussman et al. Discussion that indicates that RELN does not have a significant association with autism by any metric. You can see here, these were the genes that were significant, and this is RELN here. It does not have a significant association. So after all of this, this case control analysis and sort of this complicated explanation, we worked really hard with the experts to try to this curation. So I just want to show an excerpt from our evidence summary of the finalized curation. I'm not going to read the whole thing, but I just want to show you that we went into great detail about how the cases were statistically reported in the Sussman paper, how they found significance in de novo variation in autism versus intellectual disability, and how this wasn't supported by our current framework. We then talked about the Barrett et al. Paper and how it prompted the reanalysis by Sussman, and the reanalysis indicated that there were no significant metrics. We then described the ESAC cohort and sort of some of the pitfalls of how it's not really an adequate control population, and at the very least we should be conservative. And then we sort of outlined some of the other reasons, or we conclude by those reasons why this paper could not be scored. And then we then outlined that despite the large number of reported cases in RELN variation in CND, there's a lack of phenotypic specificity, a high amount of RELN variants detected in unaffected controls, and several lines of contradictory evidence indicating inconsistencies in statistical findings. And for these reasons, we concluded that there was no genetic evidence that should be awarded to this curation, which sort of set us up to dispute the relationship. So the last thing I'm going to go over is I'm just going to run through some of the experimental evidence and some of the inconsistencies that I mentioned earlier, as well as sort of our search to find relevant endpoints for our particular association. So the thing to note is that on this slide, we were able to score expression study studies. So this study showed that RELN was found to co-localize in the brain, which is sort of supported also by the GTEx data. So we did give a half point for expression here. We did also try to score these two functional alteration studies, indicating that RELN, I believe these were homozygous knockouts of RELN, but deletion of the RELN did cause a lack of directionality and a wandering of the neurons when they were supposed to be migrating during neurodevelopment, which may be an indication of all of our neurodevelopmental disorders, but we felt that this was better suited for the autosomal recessive curation for the lysencephaly phenotype, since that is a true malformation. So we did not score those. This next slide shows sort of 3 more expression studies as well as 2 more functional alteration studies that we scored expression, did not score functional alteration. So for the expression, we saw that the gene is expressed in mouse cortical neurons as well as the canary brain, these two. And then we also found the other type of expression indicating that RELN is actually significantly less expressed in autism patients relative to controls. So we felt that these were solid enough studies to support the association that we were trying to curate, and we did in fact score these. However, these 2 functional alteration studies, this first one showed that this particular missense variant reduced the secretion of the RELN protein, so we felt that this would be more in line with variant level evidence and could not be scored for the overall association. And then this was sort of indicating that differential methylation at the RELN promoter site. It was sort of similar to this first study. It was a study of autism patients that increased methylation was sort of correlated with the autism spectrum brains, and we felt that this was inconsistent with the Mendelian inheritance, given that it was suggesting a epigenetic type of mechanism. So we maxed out the expression, and then if we move on to the mouse models, as I mentioned, there is many studies that have looked at the heterozygous RELN mouse model. The mouse model was made by a transgene insertion that caused a homozygous knockout, and then they bred the mice to become heterozygous. So what we did score for this mouse model was one study showing that there was a decrease in exploratory drive as well as behavioral disinhibition during adolescence for the mice, and we felt that this may be consistent with sort of an autism intellectual disability phenotype. There was another study that showed that the heterozygous RELN mice had an impaired Y-maze facial recognition test, which may also indicate a level of intellectual disability. So we felt that these 2 studies, given that they did find significant sort of disabilities in the mice, we found that we could maybe score these. And there were a few other studies that suggested that learning may also be inhibited, but given that this was still the same mouse model, we felt that the mouse model itself, this specific genetic variation should only get 2 points. And to sort of support that, there are a lot of other mouse models, or not a lot of other mouse models, but there's a lot of other studies that have found sort of inconsistent findings. So for example, this study found that they did not have any significant change in the pre-pulse inhibition learning